we just say the action potential is independent of the stimulus size?
Yes. I think that works very well. What did you have, or what were you thinking of that?
I'm really novel for that one because
Novels are okay.
explain.
That's good. That's good. Actually, that's good. Good.
I had put forgot what I wrote. I'm trying to pass. This refers to the act upon reaching when on the threshold to fire. It will fire as long as it reaches a specific threshold. Anything more will not count to make a bigger reaction. Anything less will consider not fire.
Yes. I think that generally works. Did you talk about it being size invariant?
Yeah.
Okay, that works. I mean, it's not a brain buster of a question. It's just designed to be straightforward. So anything that people have along those lines will work pretty good. But that's what we're looking for. And you want to make sure that you do describe what you're talking about. A lot of activity, huh? Okay. So, all right. Any questions on that one? No. Everybody feels pretty good about it. Okay. What are the two opposing forces that drive ion movement? Explain.
Diffusion and attraction.
Come again?
Diffusion and attraction are electrical. What's it called?
Electrostatic pressure.
There you go.
Yeah. So those are the.
I just put a list. Yeah. I don't know.
Yes.
So I know that in lecture you said pressure and force. So are they? I think the book says pressure and you said force.
This is electricity.
It varies. So could it be electrostatic force, or do you want it just electrostatic pressure?
So if you just say pressure, that's not going to be sufficient because I could be putting you all under pressure right now, right? And that's not going to move your. I mean, I guess it'll move your ions, but it's not the kind of pressure that we're talking about. So again, we need a little bit more descriptive.
So electrostatic.
Yeah. So what I had in mind was
Question.
Can you go
over that
really quickly one more time?
transporters. Yes. So that was back here. So they're basically just particular receptors. They're going to work kind of like reactive pumps. So they're going to bring the transmitter back inside. So yeah, just specialized. Other question?
For the, do you think ligands or ligands?
Ligands, yeah. So who remembers what we said? So we'll walk it backwards. You have a ligand binding site. What does that tell you about a ligand? It's a thing that needs a site to bind to, right? So if we're stuck on a test and they're asking about a ligand and we can't remember what they are, right? Then you can work yourself back to say, okay, so there's a binding site for a ligand, which means a ligand is something that binds. Where would it bind to? Well, in the context of our conversation, we've been talking a lot about receptors. So that's probably a good candidate site where it would bind to, which means what binds to receptors? Usually, yeah. It's kind of what a ligand is. Yes, sir.
And then you said that, so the neurotransmitters, they don't actually go through the ion channels because they're not ions. They just act as the key so that the ion can open and let the ions through. But you said that either goes back into the cell to be recycled, or it goes out into the extracellular fluid, I guess. So when it goes in the extracellular, does it just dissolve and break down, or does it?
It can be. Sometimes something can break it down. Does anybody have an idea of what something is that might be a thing that breaks down a neurotransmitter? Esterase, indeed. Remember we talked about acetylcholinesterase? Yeah. So you can have an enzymatic degradation. Yeah. So you can have enzymatic. There you go. Enzymatic degradation. That's kind of how that could work. And if we go back to where we have our picture, that's kind of what we can have here, right? So you can have these neurotransmitters. They can be recycled out back here. They can be reuptake either by enzymes, degradation, uptake by the presynaptic cell, or by the glial cell.
No, but so something always happens to it, and it's not just going to sit there, essentially. Or what would it be?
I mean, it could sit there for a certain period of time. I mean, indefinitely until the end of the world. I don't know that it would do that. You know, I mean, it's a time issue, right? So it depends on are you clocking it with a stopwatch or a sundial?
No, because I was wondering, like, could there potentially be just like a buildup of like a neurotransmitter that's there?
Yeah, there is. That's when it's released. So that's what that buildup is. But eventually, after a period of time, it's that buildup kind of goes away, and it'll either be reuptaken or enzymatically degraded or taken in by the glial cells or cleaned out. And those things. Yeah. Good way to think about things. Other questions? Yes.
Are there instances where this process of degradation and kind of like reuptake don't work? They aren't functioning properly, so you do get like just a whole bunch of neurotransmitters there for a really long time.
Yeah, when you're on Prozac. You block the reuptake pumps. That's exactly what they're engineered to do. So, for any of you guys who don't know what Prozac is, it's what we refer to as a SSRI, or selective serotonin reuptake inhibitor. Now, serotonin, as we mentioned last class, is one of the categories of neurotransmitters, or types of neurotransmitters within the catecholamine category, that can be released to affect depolarization of this cell, right? And they tend to be involved with lots of systems, but most prominently systems, certain elements of emotion, basically affect, which is a fancy, dorky way of saying happiness. And basically, when people are depressed, they have deficits in serotonergic activity. And so this cell will release some serotonin, but maybe not a lot, or maybe not enough sufficiently to bind to these receptors to induce these cells to fire. So you have that disconnect on that system that leads to being depressed or whatnot, right? Or is manifested, it manifests behaviorally as depression, for example. So what you do with serotonin reuptake inhibitors is you can have those reuptake pumps that would be grabbing these out of the cleft and cleaning them and pulling them back into the cell. But remember, we said there's not enough of it that was released in the first place, necessarily, right? And so it's counterproductive. It's not helpful to you to have these reuptake pumps grab these, right? So what it does, it blocks those reuptake pumps. So the little bit less of serotonin that is released is able to hang out and party longer. Make sense? So it can exert more of an effect, hopefully, sometimes leading to clinical changes. Once you guys take Dr. McDougall and Dr. Amadeo's classes in psychopharmacology, but they're not the most effective things anyways. We hope that sometimes that they will lead to a change. Sometimes it can take months of taking them to lead to that change, and then you'll be lucky if you see a 30% change in 30% of the people taking it. It's not a fantastic rate. But hey, if you know people who experience those things, sometimes any kind of help is good help. You take what you get. So that's kind of the extent that we'll get in psychopharmacology for those kind of clinical disorders. It's a good primer for taking those follow-up specialized classes. Does that make sense? Okay. All right, let's see here. Okay, so we talked about some basic signaling. So here's some examples of self-signaling, okay? A knee-jerk reflex. It can be very fast, okay? And it reflects integration of both the central and peripheral nervous system. Okay, so this is just your generic thing. At the doctor's office, you get your patellar stretch reflex, okay? It's going to induce a sensory signal, okay? From our stretched receptor in our muscle. So when you hit this, it stretches your leg. It's going to send this signal. This is the sort of the dendritic side, that blue wrapping. Okay, that's the dendrite, the receptive side of this neuron. Okay, and it's a unipolar neuron. Okay, so one process comes out of the nucleus, right? That's the dendrite. Here we have the axon. That axon is going to come in. This is the spinal cord. Just a square orange box right now. But you could draw a nice little spinal cord around it if you want, a little coronal slice. But you don't need to, because it's just a conceptual representation. You remember we talked about the dorsal roots and all that kind of good jazz? Okay. It's going to have that information come in. It's going to make a synapse onto a motor neuron. That motor neuron's body is in the gray matter of the spinal cord. Remember we talked about gray and white matter? White matter is myelinated, gray matter is where the cell bodies are. That's why we know it's going to synapse in the gray matters, because it's on the cell body, which is where that is. That cell body is going to get that signal, okay? It's going to send a signal to a different muscle group that's going to contract and kick your leg up. Hit the doctor. Okay? So it's just a representation of an example of these signals being used to affect behaviors. Okay? So the behavior in this point is the motor behavior, kicking your leg in response to a particular given stimulus. And we can see these trains of action potentials, okay? So at first you have a little bit of a bump, right? That's your graded potential. Then it exceeds threshold, and you get the action potential, okay? And because stimulus intensity is encoded how? Frequency. Do you get one action potential? No. Because it wasn't a little pinprick. You got hit with a hammer, right? Not that it's a massive, massive pain, but like we've all felt it, right? So you get a couple of action potentials. It's going to transmit the information of intensity of that signal. Okay? If you measure here and then at the notorondia again, you're going to see it's the same thing. Okay? It's a little bit later in time because it's moved from point A to point B. And movement takes time. That's what we can measure in speed, right? It's not magic. So it does take a little bit. So time moves this way, so you see these trains move just a little bit over there. Okay? Now we have a postsynaptic potential here, which looks like this. Boop, 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 right? We can measure the integration of that. Okay? If this looks like it's enough to reach threshold, then it will cross that threshold, the axon hillock, and elicit a different train of action potentials that we can then measure at our axon terminal as it connects to our postsynaptic substance, in this case the muscle fiber, that we can then measure its contractions as well. So you see those series of connections? Pretty cool, huh? Anyways, let's see here. What else we got? So we've already talked about EEG a little bit. These are just macroscopic level measurements. We started off the conversation last class, starting from the high levels of EEG and breaking down the reductionism into these single neuron levels. We're just going to come up for air and take a look at how we can still see those electrical patterns of recording. So when we get those one neurons, right, firing those boop, 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 the 4 or 5 trains of action potentials we just looked at, okay, but you get one of those neurons doing that, you're never going to see EEG. This ain't going to happen. But you get millions of neurons in a community, like all in this spot, in the head or something like that. Then that electrode, C, is going to show a measurement of a trace. If they're synchronous patterns of activity. If they're not synchronous, they're going to cancel out. But then you can measure that, and that's what we're able to do. So, kind of brings it all together. And then you can see dysfunctions in that too. And you can characterize different elements of the ERPs based on when they go positive and negative and based on their temporal profile. So, just a little bit of introduction to data there. And some of those dysfunctions that we mentioned include epilepsy, seizures, things like that we can see. You're not going to have to know things about grand mal, petite, or complex partial seizures. I'm not going to test you on that stuff, so you don't have to worry about this slide in particular. But you might see, just generically, in case you're wondering if you're measuring activity, you can see normal brain activity happening, and then as a seizure starting to occur, you can see this overactivity and basically it's over element of synchronicity. So it's over synchronized. Yes.
Now, sorry, just to get during the seizure, would they actually induce the seizure to measure that? Or would they just have someone wear it and then hope they have a seizure essentially?
Well, you never hope.
I know, but like in that research, would they be like, well, we're gonna see, and if you have a seizure, we're gonna measure.
You're making a prediction that they're likely to, because they've come to your office because they tend to have seizures.
Mhm.
And so a lot of this might be actually in a hospital bed.
Yeah.
You just have them connected all the time because they just happen to have a lot of them. It's unfortunate. It's a very debilitating situation. Very frustrating for the people. It can happen to any of us, by the way. I had a buddy of mine in grad school. Just had a seizure one day out of the blue. Really freaking out. And so it's very frustrating for the people. And when they go into the hospitals, they'll get worked out. But yeah, that's how you would do this data. Is you'd either have them have with an EEG setup either at home, sitting around all day watching TV or something, just kind of doing that, or at the hospital.
Alright, yeah, I was just wondering, because I was like, that doesn't seem ethical for
Yeah, no, I mean, ethics is really important. And the other thing to mention too, right, is that when we look at that, we can see that the activity does change between different spots on the brain, right? So right frontal versus right occipital, right? You can see that, right, when you zoom out, the way you guys might just look at a quick glance, it all looks the same. But actually, look, this has a big positive peak and then it comes down negative. That doesn't happen here. This is one for a big positive arc. So the brain is acting differently in different places during the same time. It's an important part to note too, right? Because it kind of goes without saying, we kind of assume these things anyways. But we say, oh, does the brain do this? The brain do that? It's like, those are bad questions. The brain does everything. And it does lots of completely fundamentally different things at the identical time, just in different places, right? I mean, can you chew bubble gum and walk at the same time? And, I don't know, pat your head and rub your stomach? Right? Like you can do, did I do it wrong?
At first you did, I think you corrected me.
Yeah. So, anyways, okay, let's skip through these seizure things. Couple things to just mention too. Did we talk about Penfield in anatomy? So he picked up from like Sherrington's mapping of the motor cortex, I'm thinking gorillas, correct? Penfield started to do it in humans. He's a pioneer neurosurgeon. And he did the mappings of the motor cortex and lots of cortical areas during pre-surgical mapping in humans. I believe he was Canadian too, okay? And so we'll talk about him. Very big pioneer in the field. And one of the observations he made is that there is a systematic organization of the different body functions and where they map onto in the brain. Okay, so kind of bring it back home to sort of that localization structural function historically, sort of early 1900s era. And he identified something called the homunculus. Okay, some of you guys haven't seen this before, but we'll revisit. Because he found that there were consistencies in where you could stimulate in the brain of patients, and they all seemed to elicit similar responses of people. Yes?
In studies with brain patients, if they prod at certain parts of the brain, would patients be able to feel like the tingling sensation in like certain parts of the body? So let's say they're poking at one part, and the patient would say that I can feel something in my arm.
Exactly. Okay. So he found that this started to have systematicity. So you can do that to your brain, and her brain, and her brain, and her brain, and his, and they all sort of report that same feeling in that same spot that you're. It's like, okay, maybe there's a brain spot that does this. That's consistent, right? It makes sense, and it's true. Anyway, so this is an example of that motor cortex, right? So you remember we had the postcentral gyrus. No surprises there. You guys should all be experts of that. Hopefully studied it lots. Yeah. Okay. So we have that, and you could slice it. If you drew a little, I'm not going to draw with my marker because then I'll leave a bad mark on that thing. But I could just as easily push this up. That's what this is showing. It's just a zoomed in version of this. And so as you slice this apart, right, you notice that you could do exactly what you just said, which is electrically stimulate this area in just about all you guys. It's all going to move that thumb. Or make you have a feeling from that thumb. Because moving it would be the precentral gyrus, wouldn't it? Mm-hmm. Yeah. It would be more like you said, that numb feeling. Where you feel it maybe not numb, but some other sensation. And there's a systematic organization there, and look at how much cortical area in this whole somatosensory cortex is devoted to the hand and the arm. This is all just the hand. That's huge, isn't it? And some people have really big hands, but they're not. Nobody's hands are big enough to require half of the brain's sensory cortex, right? Well, it turns out all of us do. So it reflects the amount of sensory receptors in those areas too. So our hands, we'll learn about this in our sensory lectures, about all the receptors in here, because we use it as a critical way to explore our world. And we need to know lots of fine-grained detail, don't we? Right? Think about all the fine-grained details of grains of sand and everything that you can feel on your hand, different types of feeling, right? Versus like your neck or your back, your trunk, right? Like put something back here. Like I'm not gonna have a lot of sensitivity to tell you if that's a marker tip or a pencil tip or a baseball bat. Well, I'm no baseball bat. But you know what I'm saying? We'll get to this in these other lectures, but I want to introduce that concept. So this gives you sort of a quantifiable representation of the amount of neural tissue devoted to processing different sensory information in different structural areas of the brain. Okay, so it gives us then, yes.
So people that are paralyzed and they have no feeling or motor control, or if it's just they don't have any feeling due to nerve damage, would the subsequent part of that be damaged as well?
Maybe and maybe not. Depends on how that damage came or that deficit came about. So it's a complicated answer because the question can be lots of different questions actually. So if you had a brain injury to this part of the brain, then that might give you a paralysis of that, right? Which is actually what we see in stroke sometimes. So you're going to have blockage or occlusion of the oxygen flow from the brain, or from the blood vessels, bringing oxygen to some of these cells, so these cells die. So now they can't move, right? So how many of you guys have done like CPR training or basic medical stuff? You know how to identify signs of stroke? What does it look like?
Face, hands, your arm.
Oh, so you can't move either of your arms?
You can only raise one arm.
Oh,
but
why
it's
only
all
one?
balanced on one side.
Why only one?
Because their blood isn't flowing to that part.
Yeah, okay. So it's maybe this side that has those cells start to die, or they're paralyzed and can't move or feel it. Maybe the other side of the brain that is still receiving the oxygenation can move that side. That's why you have the stroke, like you said, right? You have to do one side of the face, the lateralization of these features, bringing together a lot of these concepts that we were talking about. Other times you might have a paralysis from a spinal cord injury, in which case these are relatively healthy during the injury, but it's cut in the cord, so you can't. There's no way for the signal to get from here to here, because there's a stoppage here, right? That's very different. So these cells might be fine, but what we'll learn about in lectures on plasticity is it turns out that if these, so these are your digits, your fingers, your thumb, digit 2, the bird, 4, and 5. Okay? And let's say that, and they've done this experimentally, you can go in on monkeys, and they basically chop off their fingers, 1 or 2 of them, which might sound cruel, but I mean, they'll bite each other's fingers off in fights in the natural jungles, and we need scientific research to understand what goes wrong and how do we fix it when we lose fingers in regular courses of life, which maybe you guys haven't had happen to you, but it's a lot more common than you think. I had a shop teacher in high school who was my wrestling coach, and he'd shake your hand, and he shakes your hand like this, because he chopped off the front of his 2 fingers, because he's a shop teacher. He's doing that for so many years, accidents happen. And so he grabs your hand like this, and he goes like a claw or something. So it's really, if you ever shake Mr. Tabatino's hand, he grabs it like this. And you do it before and after every wrestling match. He gives you a handshake or something. You're like, what the heck? So, but I mean, something happens, an accident. Like, how do you want the doctor to know how to treat you? All right, like we got to understand this stuff scientifically and medically. So, you do an experiment. You work off those 2 fingers with the monkey, and what you find out is that if, let's say, you lost these 2 digits over time, the space of this neural tissue will actually start to be responsive to these digits and this part of the hand will invade that territory. So there's competition in the brain for synapses. Right? This is all real estate. All right? This is like Southern California real estate. We're packed in here, right? And it's expensive. It is prime real estate down in Santa Monica or wherever all these expensive places are in Laguna Beach. You got buildings on top of buildings and hills, and everything's crammed in, and you're spending like $5,000 a month for a little shoebox, right? Okay. Prime real estate, right? You got all this neural tissue that's being crammed in evolutionarily into our higher species cranium. And so there's always competition for those resources. And so you have this property that becomes evident of use it or lose it. And so if you're not using a certain section of neural tissue, somebody else will come in and start using it. Not somebody else, but some other neural connections will start to invade there and start doing that. There will be squatters' rights. Okay? Yeah.
Is that like neural pruning?
Yeah, it's related to neural pruning. Yeah, so neural pruning isn't so much about invading different tissues. Neural pruning is about trimming certain dendrites that are not receiving connections, but the overarching concept and principle is very much the same. Yes?
What about people who feel things in like plants on limb?
That is a little bit different in that you have connections. So she asked about phantom limb. Phantom limb is if you, let's say you have a veteran who lost an arm in battle or something like that, or leg, right? The arm's gone, right? And I don't know, maybe you guys have family members or friends who might experience something like this, and it becomes this, it's sort of weird, but it's tragic too, right? It's beyond weird. They won't have the limb, but sometimes they might wake up in the middle of the night screaming pain. That arm still hurts, and it's kind of awkward because you're like, it's not there. How can it, like, right? And people thought it was this psychological, psychosomatic thing. It's not. It's actually a neurally based disorder in that you still have, so you might have lost the arm, right? Or from here down or whatever. But this stuff is still there. And so this can feel, right? In the sense that, and we'll talk about this in somatosensory lectures a little bit later, that this still receives input from neural signals. And if those neural signals happen to still get activated because they still might receive other activations, then the brain won't know necessarily what to make of it other than it's active. And we get these train of action potentials, and I interpret it to mean that it's something stabbing it or it's in pain. Therefore, we have the cognitive construction of this experience of pain, even though it's not there. It's the rewiring process of the brain. Yeah, it's very fascinating. A guy named B.S. Ramachandran really did a lot of pioneering work on that at UC San Diego. Wrote a book called Phantoms in the Brain, if you guys are ever interested in checking that out. And one way that people treat that is you can do what's called a mirror box. You set up a box, like a shoebox kind of thing that has a mirror in the middle. So let's say I don't have this arm, right? And I have a lot of pain in my right arm, right? I can have my left arm here, but then I put a mirror vertically in the middle, on the side a little bit, so that I can see the mirror image of this arm over there, which sure looks like a right arm, doesn't it? And you can kind of go, oh, there it is, and maybe even have somebody scratch it. And it just helps. Really cool stuff. Anyways, so back to what we need to do today about, okay, so there's Penfield doing some brain stimulation. Let's see, we talked about myelination, right? That propagation of the action potential, saltatory conduction, nodes of Ranvier, all things you guys should be familiar with, not in listening to, but in speaking to your friends, to your study groups. You guys should be able to communicate this stuff. And I guess I should put so multiple sclerosis is one example of when this system can go awry. So when you have the breakdown of myelin, it can lead to inabilities to move or to feel the things that that particular neuron that had the breakdown of myelin was responsible for doing. So that's what's happening in multiple sclerosis. It's generally sort of considered an immunological disorder. So the body's basically attacking itself. It's an autoimmune disorder, and it will destroy that myelin. And so remember that myelin is really important for conducting those signals. And if those signals can no longer be conducted, and what if that signal is to move your leg? What happens? You don't move your leg. What if that signal is telling you that you're touching some very soft velvet? You can't tell, or it's just numb, right? It's not sufficient enough to give you information. And so you have that poor conduction along the axons due to saltatory conduction breakdowns. Last year, I brought a patient in who had multiple sclerosis at the end of the semester, I think after finals. And he talked about his experience with it. He was a roommate of mine in graduate school. He got hit with it probably about 4 years old, and had a very interesting ordeal with it, and talked about the medical treatments that he pursued, and it's actually in remission. So there's some new experimental treatments using stem cell therapies.
For MS?
Yeah. Yeah, it's really pretty incredible. Kind of miraculous, you might say. So he had a lot of work done at Northwestern University that he was an experimental test subject for, and it basically put it in remission. So you do chemotherapy and basically destroy your immune system and your and then you rebuild it, and you use some stem cells, and he has grown a new immune system. Yeah, it's pretty incredible. If you guys want, I can try to bring him in again this year. Maybe after finals again. It's not really enjoyed a lot more class time out right now, but maybe if you guys are interested, I can have him come back in again, maybe lunch or something. Very interesting story. So he found a lot of experience of it with diet and food that he found helping his recovery, and that he's looked into the literature on. But right now it looks like the real factors driving a lot of that is the stem cell replacement therapy. So very interesting stuff. Looks like we only have a minute or two left. This is showing that degradation of myelin here, and why you might imagine that muscle might no longer be able to move. And again, we talked about tingling sensations, all sorts of things that the neurons are responsible for seeing things is going to go bad. So you're going to have blurred vision. Motor and sensory things that neurons communicate information for, that's going to go bad too. You're going to get some of that confusion. Coordinating walking, that's part of that motor coordination stuff. So what I want to finish with is one more thing, and then we'll let you go on our synaptic transmission. Let's see here.
Bio kids, put your books down.
Maybe it's a way to help study and remember some of the synaptic clefts.
Synaptic cleft. I believe Bob Voltage. Introducing the Gluten Clan. Hot and turns to the lane of the nerve where firing and wiring will occur. Follow me, neurons gotta be ready to fire at any opportunity. Dendritic input, make it hot. Sum it up in the action hill lock. The potential will rise to a constant size. Shift the action potentials, ain't no surprise. Snap back to the focus of the rap. Tiny little space, aka synapse. Voltage switch to the end like a blue. Calcium rushes in, vesicles go boom. So psychosis, so exciting. ACS spines light it up like lightning. Snap fanatics gather around, questions about the story. Do the transmitters always excite? Now they can be inhibitory, depending on whether they're the ions of K rushed up, positive charge gone. Only one synapse? What if there's more? The whole life will sum it up like I said before. The synapse is the location for neuron to neuron communication. Remember what Russ Fernald said. Mind arises from the brain.
Check out the synaptic clefts, thanks to vesicular tract.
Ion channels.
Neuronal signal can be transmitted from electrical to chemical and back
Ion
again.
channels. Next to the synapses with exact like capsules and chemicals
that make you relax or collapse. The synaptic flex has receptors, the box of the synthetic ligands live in my clinics that mimic no chemical condition. That's a small problem for medicine. 100 trillion synapses, drug screaming. Medicine! For disease is only one part of the brain. Well, pills not a sniper, it's a hand grenade. It'll act wherever there reside defects, plus everywhere else comes side effects. Don't wanna sound like a cynic, we've all got a protein they call nicotinic. It's a receptor that can't tell the difference between ACH and nicotine. Get a grip, but nicotine leads to too much binding. So ACH receptors get to hide. Neuromuscular junction, it can't quite function, cause your receptors were now combustion. Now dopamine's on for reward prediction, but it's down regulating and getting addicted. Mixing the kick to habit with busted synapses. How trash synaptic receptors getting abducted? A healthy synapse ain't nothing to muck with. I hope this rap has been instructive, cause a healthy synapse ain't nothing to muck with.
You don't have to read it.
Alright, that's pretty strong wrap up for today. If you guys who liked it, and maybe an easy way to help prepare some stuff. And there's nothing stopping you guys from making your own videos too, you know?
My mom's
Oh, you guys want to see one more thing?
5 a.m. Yeah.
Here. This is sodium potassium pump. Seriously, you have the extracellular, the side is intracellular. You have ATP or energetic source transferring 3 sodiums for 2 potassiums.
Oh, that's cute. I like that.
I'll see you later.